right, we're back. That foundation we just developed, the story we just told about how the genetic influence and the environmental influence combine to establish normal distributions of quantitative traits, is a nice story. We like to call it a nice theory. But is it true? That's obviously an important point. We can't really see exactly what's happening under the hood directly. We can a little bit better today with genomic data, as we'll see in a subsequent lecture or two. But even now, our view of it is rather murky, so we have to subject the theory to other kinds of indirect but clever tests. And the classical way of doing this, as soon as the ideas were born, as I said, in the early 20th century, a little over 100 years ago, was to do breeding experiments. And they were and remain very convincing. Here's a picture summarizing a famous experiment done by Edward East in the early 19-teens. He crossed purebred lines of tobacco. You remember this from our little cartoon of increasing numbers of loci. So here's the actual experiment done on these inbred lines of Nicotiana longiflora that differed in corolla height, that is, on how longy their flori were. It is obviously a self-compatible hermaphroditic flower that's happy to mate either with itself or with another individual. That's how he made these inbred parental lines with notably short and notably tall flowers. The F1s were intermediate, as we said, and there's a histogram showing their distribution on this scale, which is millimeters at the bottom. But they were not notably more variable than the parentals, an important point to which we'll return. The F2s were also intermediate, but they were more variable, and that's a prediction of the model. Their variance should go up since they will have a range of different genotypes, as we saw in the cartoon story of the model, and sure enough, they do. The F1s are heterozygous at every locus where the tall and the short are fixed for a different allele, so we expect their variation to be purely environmental, and it is pretty similar in magnitude to that of the parents in this experiment. What he then did was breed selectively from the smallest flowered and the tallest flowered ends of the F2, F3, F4 generations to finally produce an F5. He was hoping to reconstruct, or to see how close he could come to reconstructing the parental distributions, and he was making progress, seemingly especially with the short flowered end of the spectrum, not so convincingly with the tall flowered end, interestingly, but he was getting there. He had created a complete gap in the middle by then, lacking intermediate flowered individuals. So the implications of that were that many polymorphic loci did contribute to corolla length in this population and species. Otherwise, he would have been able to immediately reconstruct the parentals. And there is also environmentally induced variation, even among the genetically identical parental lines and the F1s, which have less variation than the generations that are still apparently segregating for many of these plus and minus making alleles at different loci, but that is important, significant quantitatively. A modern way of testing whether the theory is true is to try to go find, that is by genetic mapping, these quantitative trait loci, which are often abbreviated QTLs in the trade. Here's a nice example from a real wild species, monkey flowers, mimulus, which have several species in North America, including this interesting situation where a group of very closely related species, especially these two, Mimulus cardinalis and Mimulus lewisii, differ dramatically in the color and morphology of their flowers because they are pollinated by completely different organisms. Cardinalis is hummingbird pollinated, as are these more distantly related species that also have red, laterally flattened flowers. But lewisii, a close relative of cardinalis, so close they're fully interfertile, has a classic bee-pollinated type floral morphology, a more radially symmetric open flower that bees can approach and work with easily. Bradshaw and colleagues said, all right, what's causing this difference in flower morphology and other traits that are related to attracting hummingbirds on the one hand and bees on the other? How did that difference evolve? That was their question. Okay, so they crossed cardinalis and lewisii and then made large numbers of F2 progeny by crossing the F1s. So here's an actual photo of a typical lewisii flower close up. Looks like a bee-pollinated flower. And here's cardinalis. Looks like a hummingbird-pollinated flower. Here's the F1, amazingly halfway in between the two. And then here's a group of 9 F2s carefully selected for the degree to which they approximate the lewisii condition, which this lower left-hand one pretty much does, and the cardinalis look, which this one in the lower right does, but there are lots of F2s that are still a mix of parental traits. Okay, so now to locate the quantitative trait loci, the hypothesized quantitative trait loci, what do? You correlate linked marker genes with trait values. So what they did on the F2s was score 12 different floral traits. Here's a list of them. It includes the flower morphology traits. It includes nectar volume, which is a reward for the pollinators. And it includes these dimensions that affect how efficiently the pollinators actually can pollinate. And then they looked for associations among the F2s between parent-specific genetic markers and the trait values. So they had a bunch of loci. I don't know what they were. They might have been microsatellites. Nowadays they would be SNPs. And at which the two parental species were homozygously fixed for different alleles. So MC means homozygous for the cardinalis marker, and ML is homozygous for the lewisii marker, and of course MCML would be a heterozygote for one of each. And so the test is that to look for markers that seem to be affecting the trait. All right, so that indicates that the marker is probably not the quantitative trait locus itself, but that it is linked and is in linkage disequilibrium with the quantitative trait locus for the obvious reason that it's been in a separate population for a long time. Right, so in cardinalis, many of these cardinalis marker, cardinalis QTL pairs got put together, and likewise in lewisii marker alleles got paired up with lewisii alleles at the quantitative trait locus. And then in the F1s, we create instant linkage disequilibrium, maximum possible linkage disequilibrium, and then we follow how those pairs segregate out in the F2 and perhaps succeeding generations, but in this experiment they just work with the F2s. And what they found was a goodly collection of linked markers that did predict effects on the phenotypes of interest, these 12 traits. And so these histograms just show the percent of variance explained by various of the quantitative trait loci that were formally identified by their linkage with markers. And some of them explained amazingly large fractions, like 90% of variation in one of the attraction traits, and many explained substantial fractions of the phenotypic variance in the other traits as well. The reward trait, number 8, nectar volume, and these efficiency of pollinator manipulation dimensions. So that was all good news, and confirms, as many similar experiments have, and we'll see some examples of others, modern linkage studies, so-called genome-wide association studies that we'll come back to, probably not next time, but in the last lecture next week. And so we do believe that in general outline, this model still, it seems as convincing as it did 100 years ago, in many ways more so, and as we'll emphasize when we get there, raising as many interesting questions still unanswered as it answers. In any event, what we do believe we know is that indeed quantitative traits do vary, and they are always normally distributed, and offspring tend to resemble their parents. Here's a graph of mid-parent beak depth on the x-axis in millimeters versus offspring bill depth on the y-axis in Darwin's medium groundfinch, which we saw cartoons of at the beginning, for two separate samples on the same island with the same population before and after a big drought in 1977 that selected for larger body size and beak dimensions. This implies, these observations imply that some of the variation is genetic and hence evolvable, as it was in this famous example from Darwin's finches. Here's the joint mean of parental and offspring phenotypes, that is beak depths, before the drought, and here it is after the drought, where it's been shifted upward, both in the parents and even more so in the offspring, lifting the regression line for 1978. Yes, the post-selection population line up. That part is actually an environmental effect. The genetic effect is basically this movement of the mean in the left to right direction. The conditions were terrific in 78 for raising offspring who got many more seeds because there were fewer birds around to compete for them when the plants got happy again, when the rains returned and started making seeds. So breeding experiments in many, many species, and both models and in the wild, like this Darwin's finch case, suggest that the genetic contributions do indeed come from genotypes at least several and often very many, sometimes very, very many loci. And as advertised, effects of the environment contribute a blur to the effects of genotypes. Okay, and I believe that's it. We will see you next time.